Welcome back to G-Fun Facts Online. Today, we are going to change the way you look at the fundamental building blocks of life. We are usually taught that DNA is a script, a code, a biological software that dictates whether you have blue eyes or brown, how tall you grow, and your susceptibility to certain diseases. But what if I told you that for the last 40 years, a group of scientists has been ignoring the biological function of DNA entirely? To them, DNA is not a code. It is a brick, it is a girder, it is a programmable construction material. This is the story of programmable DNA matter, and how we are currently standing on the brink of a new industrial revolution, where we don't build things with hammers and drills, but where we program matter to build itself. To understand how this started, we have to go back to the quiet, sterile labs of the 1980s. There was a crystallographer named Nadrian Seaman. He wasn't trying to cure cancer or map the genome. He was wrestling with a very specific, structural problem. He was trying to arrange proteins into orderly crystals so he could study them. Imagine trying to stack marbles that refuse to sit still. It was frustrating, meticulous work. His solution came from a moment of inspiration in a campus pub. He realized that the molecule famous for carrying the code of life, DNA, could be repurposed. He realized that the strict rules that bind DNA strands together could be used to force molecules into specific geometric shapes. This moment birthed an entirely new field of science. 40 years later, we are no longer just reading the code of life. We are building with it. Welcome to the era of programmable DNA matter. This is a frontier where biology meets engineering to create self-assembling 3D nanostructures. This is not science fiction. Today, researchers are programming strands of DNA to fold themselves into tiny robots that hunt cancer cells, nanoscale factories that assemble molecules atom by atom, and archival storage drives that could hold the entire internet in a shoebox. So how do we turn a biological molecule into a construction brick? To understand this, we have to look at the architect's blueprint. We need to look at Watson-Crick base pairing. DNA consists of 4 bases: adenine, thymine, cytosine, and guanine. We call them A, T, C, and G. In biology, the sequence of these letters determines your genes. But in DNA nanotechnology, we only care about the binding rules. The rules are strict and unbreakable. A always pairs with T, C always pairs with G. This predictability is an engineer's dream. It means that if you synthesize a strand of DNA with the sequence ATCG, you know with absolute certainty that it will bind only to a strand with the sequence TAGC. By carefully designing sequences that complement each other at specific points, scientists can program DNA strands to self-assemble into complex 3D shapes. Think of it like designing a Lego set, where every brick has a specific barcode and magnets that only snap to the correct partner. The beauty of this is that you don't build the structure manually with microscopic tweezers. You simply mix the bricks in a test tube, heat them up and let them cool. As they cool, they frantically search for their partners, spontaneously forming the exact shape you designed on a computer. It is self-assembly in its purest form. Currently, there are 2 primary techniques dominating this field, and they represent 2 different philosophies of construction. The first is called DNA origami. Invented by Paul Rothemund in 2006, this is the technique that really put the field on the map. It uses a long, circular single strand of DNA, often taken from a bacteriophage virus. This long strand acts as a scaffold. Then, researchers add hundreds of short, synthetic strands called staples. These staples bind to specific, distant regions of the long scaffold, pinching and folding it into a compact shape. It is exactly like taking a single sheet of paper and folding it into a crane, except the paper is a molecule, and the hands folding it are the laws of thermodynamics. This method is incredibly robust. Scientists have used it to create microscopic smiley faces, maps of the Americas, and even tiny boxes with lids that open and close. The only downside is that the size of the structure is limited by the length of that initial scaffold strand. The second technique is called DNA bricks. If origami is paper folding, DNA bricks are true Legos. Developed by groups at Harvard and the Weiss Institute, this method gets rid of the long scaffold entirely. Instead, it uses hundreds of short, interlocking strands that bind to 4 neighbors each. This allows for modular designs. You can build a massive cube and then carve out a sculpture by simply removing specific bricks from the mix. This allows for the creation of 3D canvases with thousands of distinct volumetric pixels, or voxels. While this method originally required very strict conditions to assemble, recent techniques known as crisscross polymerization have made it much more robust. Now, let's move from theory to application. The most immediate and life-changing application of programmable DNA matter is in medicine. We are currently moving from the age of dumb drugs to smart nanorobots. Think about chemotherapy. It is a chemical carpet bomb. It floods the entire body to kill a few bad cells, causing terrible side effects. DNA nanorobots offer a different path. One of the most famous examples is a DNA machine designed to kill tumors. These are essentially DNA clamshells. Closed tight, they travel harmlessly through the bloodstream, but they are equipped with a lock mechanism made of DNA-AP tamers. These are short strands that recognize specific protein markers found only on the surface of cancer cells. When the nanorobot bumps into a healthy cell, nothing happens. The key doesn't fit. But when it bumps into a cancer cell, the cancer marker fits the lock. The clamshell springs open, exposing its payload. This payload could be an antibody or a coagulation drug that cuts off the tumor's blood supply. This ensures the drug is delivered only where it is needed. It is a magic bullet in the truest sense. Beyond killing cancer directly, these structures are being used as an artificial immune system. Researchers have built 3 ddNA tetrahedrons, which are 4-sided pyramids that act as rigid scaffolds for presenting antigens to immune cells. Because the spacing of these antigens can be controlled with sub-nanometer precision, these structures can stimulate an immune response far more potent than free-floating proteins ever could. It is a vaccine, engineered to architectural perfection. While biologists are using this tech to look inward, material scientists are using DNA to build the external world. They are performing alchemy at the nanoscale. DNA is being used as a template to organize inorganic materials like gold, silica, and quantum dots into structures that nature simply cannot produce. This leads us to the concept of metamaterials and invisibility. By attaching gold nanoparticles to precise points on a 3D DNA crystal, scientists can create optical metamaterials. These are materials with structures smaller than the wavelength of light. This allows them to bend electromagnetic waves in unnatural ways. Theoretically, a perfect metamaterial could guide light around an object, rendering it invisible. A real-life cloaking device. While we aren't there yet, DNA scaffolding provides the only viable path to manufacturing these complex 3D lattices at scale. Another application is molecular lithography. Computer chips are currently made by carving patterns into silicon with light. But we are hitting the physical limits of how small we can carve. DNA offers a bottom-up alternative. Instead of carving down, we can build up. DNA bricks can self-assemble into a template for carbon nanotubes or metallic wires, creating circuitry that is smaller and more complex than anything Intel or TSMC can currently etch. Then there is the data crisis. Humanity generates about 2.5 quintillion bytes of data every day. Our magnetic tapes and hard drives are running out of space, and they degrade after a few decades. DNA is the ultimate storage medium. It is millions of times denser than a hard drive. If kept cool and dry, it can last for hundreds of thousands of years. Just look at the DNA recovered from woolly mammoths. Programmable DNA Matter allows us to move beyond simple sequence storage. Researchers are now building 3D DNA files. Imagine a microscopic library where data is not just a string of letters, but a physical structure. By using nanopores, which are tiny holes that read electricity as molecules pass through them, we can read the shape of a DNA nanostructure to retrieve data. Companies like Catalog and Iridia are leading this commercial vanguard. Catalog, for instance, isn't synthesizing new DNA for every bit of data. That would be too slow. Instead, they use a library of pre-made DNA words and stitch them together into massive sentences using enzymes. It is essentially a printing press for genetic data, drastically lowering the cost of storage. Now I want to talk about the very edge of the frontier. In late 2024 and throughout 2025, the field witnessed a breakthrough that sounds deceptively simple. It is called DNA and water self-assembly. Researchers at Columbia University and Brookhaven National Laboratory developed a method where DNA strands are attached to soft particles, which then act as voxels. By tuning the DNA strands to repel water in specific patterns, they force the particles to arrange themselves into complex macroscopic crystals. This is a paradigm shift. It means we aren't just building wireframe DNA shapes anymore. We are building solid, volumetric matter. It is the difference between drawing a cube on paper and holding a wooden block. These DNA-programmed materials can switch properties on demand, becoming conductive, transparent, or rigid when exposed to specific triggers. So if this technology is so powerful, why can't you buy a DNA computer at Best Buy? There are 3 main challenges. First is cost. Synthesizing high-quality DNA is still expensive. While the price has dropped from dollars to pennies per base, building a gram of DNA nanostructures is still exorbitantly pricey compared to plastic or silicon. Second is the error rate. Self-assembly is a statistical process. Most bricks find their partners, but some don't. In a drug delivery vehicle, a 1% error rate is acceptable. In a computer chip, it is catastrophic. And third is physiological stability. Your body hates foreign DNA. Our blood is full of nucleases, which are enzymes evolved to shred viral DNA. For medical applications, DNA nanostructures must be coated or chemically modified to survive the journey to their target. Despite these challenges, we are standing on the precipice of a molecular industrial revolution. Just as the ability to forge steel defined the 19th century and the control of silicon defined the 20th, the control of self-assembling programmable matter will define the 21st. The vision is grand. Scientists envision universal fabricators. Imagine a vat of liquid containing trillions of DNA bricks. You download a schematic for a smartphone, a vaccine, or a solar cell. You inject a seed crystal into the vat, and before your eyes, the liquid clouds over as the structure grows, atom by atom, into the final product. It starts with a single strand, finding its partner in the dark, but where it ends is limited only by our ability to write the code. Thanks for listening to G Fun Facts Online.